I am Carmen Lupascu. I'm a postdoc in Michele's Migliore's lab, and I will going to introduce the main algorithm that we are using for the single cell optimization. So we have seen yesterday that what's the pipeline for the single cell optimization, and now we will talk about how to optimize the mathematical model against the experimental constraints. It will be a very technical presentation. I'm going to talk about the optimization algorithm, which is called Bluepieopt. Was written by Werner van Geet and al. And you can, it's an open source algorithm, so you can find it on GitHub. And we will see what algorithm it uses, what's the simulator, the feature extraction you have seen already yesterday, and how the parallelization is done. So Bluepieopt is a multi-objective evolutionary algorithm that relies on a Python library, which is called DEEP, Distributed Evolutionary Algorithms in Python. And it provides many algorithms, such as covariance matrix adaptation, particle swarm optimization. And to define a Bluepieopt optimization, the user must provide an evaluation function that maps the model parameters to a fitness score. And Bluepieopt can interact with the following external simulators from within Python. So Neuron, Nest, Pine, Brian, Steps, and Moose. You have seen yesterday that for an evaluation function to compute a fitness score from the simulator output, the resulting traces must be compared against experimental traces. And the Blue Brain project has released the FL library, so the electrophysiological feature extraction library, which is also an open source software. For the parallelization, the DEEP provides an easy way to evaluate individuals in a population on several cores in parallel. And the user must provide a map function. So it can be a Python serial map in the standard library, or it can be a parallel map function in the multiprocessing module to leverage local hardware threads. And for parallelization over a large cluster, the DEEP developers encourage to use Scoop. So the optimization algorithm, Bluepieopt, uses a general indicator-based evolutionary algorithm called IBEA. And like any genetic algorithm, it has an initialization step. So a population of size n is generated at the beginning. Then for each individual in the population, a fitness value is calculated. And if it doesn't reach the stopping criteria, it does a selection first. So tournament selection is performed with replacement on this population to generate a temporary population. And this population, it reproduces using genetic operators. And a new set of individuals are generated. And then using environmental selection, so the new individuals are inserted in the population, and the fitness values of the individuals are iteratively updated. And at the end, the worst individuals are removed until reaching back the original size of the population. And this goes on iteratively. The software architecture, Bluepieopt follows an object-oriented programming model. So the various concepts of the software are modularized into classes. There are some classes like model, morphology, mechanisms, protocols, stimuli, recordings, and location that are specific to set up neuron models and assessing their input-output properties. And there are other classes like objective and e-feature that are more generally applicable So for example, the EFL feature class provide feature extracted from voltage traces using the open source EFL library that we've seen yesterday. At the highest level of optimization, the Bluepieopt contains the classes optimization and evaluator. The evaluator object defines an evaluation function that maps the parameters to the objectives, and then the optimization object accepts this evaluator as input and runs a search algorithm on it to find the parameter values that generates the best objectives. The goal of the algorithm could be the minimizing of a weighted sum of these objectives. There are some other classes. For example, the protocol class can attach stimuli and recordings to the model, and when the simulation is then run, a response is generated for each stimuli for a given set of model parameter values. Another class, location, is created in order to specify the location on a neuron morphology of a recording, stimulus or parameter. And then the objectives calculator class takes the response of a model and calculates the objectives values from it. The code, so we'll talk about the code mainly. You can find the list of some optimizations that can be used also later on the hands-on session this afternoon. I will ask you to download this repository, or at least one folder of this, in order to see what's the structure, maybe if you want in the same time as I'm doing the presentation. Because this code for setting the parameters, objective and optimization algorithm is partitioned into separate modules, okay? So when you want to download at least one folder in the GitHub repository, you will see that it has the same structure that I'm describing now, okay? So there's a config folder where in generally configuration values are stored in this and read from this JavaScript object notation, so JSON files. In this configuration file, configuration folder, you will find a file named morph.json, where the morphology used in the optimization is specified. In this case, we are using an ASC file for the morphology. And then to compute the model score that will be used by the optimization algorithm, we defined objective objects. So in the features.json file, you have seen already yesterday the structure of this JSON file. You can find this again in the config folder. You will see there's in the model folder there are this Python file. This is called evaluator.py, and it reads the JSON file in order to define the fitness of this objective. So the recording names here takes as an argument a dictionary where the keys are the recording location. In this case, this is an empty string, and it refers to the soma, okay, the recording location. Okay. How it's computed, how the objectives are computed. So for every feature value, one objective score is calculated following the Druckmann et al. Formula. And this approach normalizes all objective score to a common scale, allowing them to be combined regardless of the units, and weights specific objectives according to feature variability. Going back to the config folder, there's a protocols file where the protocols are specified, and again there's a function reading these protocols.json And the step amplitude argument of this neuron square pulse attribute of the IFIS class corresponds to the amplitude of the current pulse, like here in the JSON file. Yes?
In your previous slide, there were two numbers for each of the features. One more.
Yes.
Is that an allowable difference?
It's mean and standard deviation. Just mean and standard deviation of the feature. This step amplitude argument just specifies the amplitude of the current pulse, and the step delay, step duration, and total duration, like in the protocols.json, specify the start, the time, the length, and the total simulation time. Okay. Last file in the config folder, there's parameters.json. So this is the main file to specify the parameters to be optimized. There are some parameters marked as fixed, and these are kept constant throughout the optimization. And we specify here a range for the parameters to be fitted, and the distribution of these parameters within this range. The location of the parameters is based on the usual section list names automatically assigned by neuron as somatic, axonal, apical, and basal section lists. The mechanisms are specified as mod files and of course combined with the parameters.json. These are appended as mechanisms to the IFIS object. Okay? Once we created a cell template, and we see above here how to create a cell template, to optimize the parameters of the cell, we need to create a cell evaluator object. This object needs to know, as we have seen before, which protocols to inject, which parameters to optimize, and how to compute a score. And the cell evaluator constructor has a field, param names, here, has this field param names, which contains the list of the names of the parameters that are used as input and that has to be fitted. And this is the ordered list of names. Okay? And finally, the main Python file that you can find in the folder, it's optneuron.py. So finally, once we have a cell template and evaluator for this cell, the deep optimization object can be created and run. And as I told you before, the deep algorithm is an evolutionary algorithm, so it evolves a population through consecutive generations. And for each generation, we must set offspring individuals, and these are generated from the selected parents from the previous generation. And we can specify the size of the offspring population and the maximum number of generation when we are specifying the deep optimization object. Once the optimization is run, classes are provided to keep track of the Hall of Fame of individuals, is called, during the evolution. So there are some recordings of population statistics in a such called logbook, and the genealogy between individuals can be saved, analyzed, visualized. This is so-called checkpointing, and it's implemented in deep by storing the algorithm states in a Python pickle file for any generation. And this can be read using a Python coding that can be easily used also in the collab in a Jupyter notebook. So here we can see that the keys of the pickle file are the generation, the Hall of Fames, the parents, the logbook, the population, and the history. And for this specific PKL file, we may see that there were just 2 generations in the optimization. In the Hall of Fame, there are saved only the 10 best individuals, usually. And we can see that each individual was 22 length, had a length of 22, so maybe, of course, 22 parameters to be fitted, okay? Inside the checkpoints folder, we can find also a Hawk template that contains the best parameters after the optimization, and it contains the morphology to be loaded. Sorry. Okay, this is the presentation. I cannot go back to see the structure of this file, but it's for those of you that know Python, Neuron, it's a normal template structure of a cell. And finally, for the analysis step, so after we get the results of the optimization, we can run an analysis step, and inside the figures folder, you can find these 3 types of figures. So in the first figure, you can find the traces generated by the best solution in the Hall of Fame. And of course, the best solution has the objective values that are in the same range as the reference model. Then you can see that the objective scores for the best objective values found by the Bluepieopt. Of course, the goal will be to minimize these values. And finally, we can see how the optimization evolved. So we can see here the minimal, the maximal, and the average scores found in the consecutive generations of the evolutionary algorithm. So here we can see that 60 generations were used.
Okay, my name is Stefano Masoli. I'm a postdoc in Egidio Dangero's lab, and I will go on talking about Bluepieopt. I will give you some example of what I'm doing with it. My approach is slightly different than the one already seen, because for me, biology is about reusability than standards. You cannot find something standard in biology, something that can be reused by the system. A channel is used in a cell, can be transported in another cell and have some other effect. Or, for example, in this case, all neurons have dendrites, soma, axons, but they are different. For example, if I take a Purkinje cell, it has a huge dendritic tree with many sections, different diameters, different lengths. But if I take Granule cells, it has only 4 dendrites and a small axon. But if I take another cell, an entirely different cell, for example, a pyramidal cell, is even bigger than a Purkinje cell. But it has a different distribution of the dendrites based on the diameters, the length, and so on. So each cell is like a different, even is equal to the other. So starting from the basics, the section list, which already has been declared. The original one used by Neuron, defined by Neuron, are somatic, axonal, basal, and apical. So every time I find a morphology about one cell, if I load it up into Neuron, I can obtain the division of all the section based on somatic, axonal, so axon, dendrites, into this section list. This is in according with the type of morphology. So if the morphology, for example, an SWC format, which is the most common in the end, I have to set 1 for the soma, 2 for the axon, 3 for the basal dendrites, and 4 for the apical dendrites. But this is not something that can be easily done, because many morphologies lacks axon, so there is something that is missing. Many other morphologies have only basal dendrites, so there is no distinction between basal and apical dendrites. So if I want to optimize that cell, I have to take in account that it has only basal dendrites, or it has apical dendrites too. But maybe the one that has done the morphology wasn't aware of that. So the same with the neurolucida format. One can define axon, dendrites, basal and apical, and the cell body. So it depends on the morphology. to have the basic section list. 3 years ago, I requested to Werner van Geit, that is the author of Bluepieopt, to be able to define my own section list. So I can define something like actual name of a variable like all, trunk, axon initial segment, and so on. Why? Because I was in need to have something that I could play with. So, for example, placing the specific channels in a specific location that the basal and apical were too strict for them. So I can create a malinated axon. I can do a multi-branched axon with the initial segment, and so on. So you can combine them. You can have the default section list plus a custom section list you can create. You have a higher degree of freedom. Why? Because sometimes a cell looks like this one. This is the Purkinje cell. It's massive. So to give you a figure, this is from mouse. But if you take one of us, so it's a human Purkinje cell, it's 1 millimeter for 1 millimeter. It can be seen without a microscope. So it's so big that all the morphology I found, they define only as basal dendrites. As you can think, it's impossible to define a channel starting from the soma to the apical as a single layer of complexity. It's impossible. In fact, many experiments were done in the last 3 or 4 decades to define that the cell has specific location for specific channels, which are not possible to be defined only with a basal and apical approach. So in this case, this is the original from a guinea pig. So I have a big, massive dendritic tree. Dendrites were divided by the diameters. So it's that is the reference. I've added the axon initial segment, paraxon initial segment, nodes of Ranvier, axon collateral, and everything. So this is the model I've done in 2015, published in 2015, and it was done by hand. So manual fitting of each single conductance on the morphology. And this looks like a mess because all the channels are divided based on literature. So there is at least one paper that says one specific channel has been recorded or seen with immunochemistry in that specific location. So I have 3 different layers in the dendrites. I have what for me are the apical dendrites, the first. So everything below 3.5 micrometers of the diameter of the dendrites is apical. The rest is like basal, is the main trunk. And something that is specific for the Purkinje cell is that other cells have sodium channels in the dendrites. In this case, there are none, except in the first part of the trunk. And for the rest, the soma, initial segment with specific channels, para-initial segment, which is a very strange section, node of Ranvier, and so on. So the idea was to be able to do the same thing I've done with Python and Neuron by hand, doing the same in the Bluepieopt. So I have to request this modification even in this case, because this is the initial segment that is now the most important part of each neuron on the planet, because it's the center that generates the action potential. So without it, the cell is dead, more or less, as we'll see. In the case of the Purkinje cell, without the initial segment, is dead completely. So the concept is having a small section, which is around 17 micrometers long, with the sodium channels, potassium channels, and 2 types of calcium channels. One day, they published a paper about motor neurons, and they found out a specific small section at the end of the axon initial segment and before the myelination, which is called para-initial segment. It's filled with potassium channels, in this case Kv1.1, which is very, very low threshold. So it's like acting like a dampener. Everything that is generated in the initial segment can travel through the axon. Everything is generated in the axon is damped, so it's not interfering with the actual action generation. Of course, to do that, I had to request a modification to the original code of Bluepieopt, because the original class was unable to sustain more than 2 sections for the axon, for example. So this is a class that overwrites the original one present in the packet that you can install from GitHub. is based on the fact that I declare a new class with a new name that super, so goes over the other one that is AFIS morphologies and error file morphology. With this definition, I can really create anything I want. I can define any section list. I can do anything I want. For example, in this case, as you can see, axon 0, axon 1 is the initial segment and the par-initial segment, and the original code goes on with first manual section, node of Ranvier, and so on. It's a long list, is over 12 lines long. So if you want to take a look to what I've used as an example, as a starting point for doing an optimization, on GitHub there is a directory with an example, which is simple cell. It's very linear, 2 channels, nothing else. And that was the basic for my version of the code, because it can be written, like told by Dr. Lupascu, can be defined by JSON files or directly in the code, like Python. It's a different approach, the results is the same in the end. So in this case, I define section list for specific location. So then it means the dendritic section of the entire morphology. Then trunk are only the dendrites of the main trunk. Then sodium are the section only for the first part of the trunk. To add section list is very straightforward, because one declares what the name of the section list, where to use it, so then the whole section list name, etc. And in the previous class, one has to define the moment when you create the section list. So for example, in my case, I have access to isCell.dend, which is the list that contains all the sections. And from there, I define simply, if the section is between, the diameters between 0 and 12, I do an append to the section list that contains all the sections. If it's between 2 other ranges, I add only those specific sections to that specific section list. And the same can be done with, for example, the axon. So I create a section list for the initial segment, and I can do all the rest of the axon. This permits, for example, to have a morphology with the axon, maybe a multi-branched axon. I can define the section list for the first section, so I define the initial segment, and all the others are loaded by the program without intervention. So it can be done some mix-up of the two approach. After doing the optimization, I obtained a Purkinje cell. In this case, Purkinje cells are known for being very regular, so the spontaneous firing. In this case, it was around 50 Hertz. If I do a current injection, I obtain a much higher frequency, again, accordingly to the one I've done as a manual fitting. So this is actual results from an optimization. Now, this is one of the slides that I've used for the validation. Now, it seems a strange graphics because the cell is dead. That is why the initial segment is important, because if I took out the initial segment, as has been done in experiment, the cell is unable to fire on its own. And this is my key point for the validation, because, okay, I can do an optimization, I can obtain results, but if the results are not correct, what's the point that I've done the optimization? So the most important critical part is the validation of the results. This is my validation. So if I take out sodium channels from the initial segment, the cell is still able to run? No? Okay. Yes, it's wrong. The other one is the complex bursting. It's something that is not usually seen in reality, because it's a very extreme current injection. But if you do that on mice or a guinea pig or something, you do a current injection over 2 nanoampere, you obtain something that is called complex bursting, because it's a calcium spike followed by a sodium burst. If the optimization was wrong or was unable to find the correct conductances for all the channels, this behavior cannot be seen. It's like a test bench. If a cell is able to reproduce both of these properties, then the cell is an actual Purkinje cell. And this is something that I'm still working on. The concept is I have many different new Purkinje cells. This is from mice. For example, the one on the right is only 80 sections. The other one is 400 sections. Still Purkinje cell, taken from the same slices, taken from the same animals, one attached to the other, literally. With the same section list, I'm able to obtain the same, or at least a similar physiological activity without changing anything else. So the morphology is different. The results are equal, or at least there are some variation in the frequency, but amplitude, spike shape, that is very important, and the overall response to the current injection are the same. So ranging from one side to the other, the granule cell, this is the other example. It looks like a simple cell, it isn't. It has only 4 dendrites, usually 15 micrometers long, a very small soma, and a very long axon. The ascending axon can be long even 500 micrometers. The problem is the parallel fibers, because the axon splits into 2 parts and generate an axon that can be long 3-4 millimeters. So it's very, very long and very thin. Usually it's around 0.15, something less, something more, but that is the average. The importance of this cell is that they receive the information from outside the cerebellum, they start the processing, the elaboration part, and then send the information to the Purkinje cell. So they are interconnected. One granule cell can contact thousands of Purkinje cells. So in this case, I used this custom section list in a reverse way. Instead of using to build a small section of the axon or play around with the dendrites, I generated a small section of 7 micrometers long, repeated. So for example, I have an extending axon that is composed by 18 sections, or in another case, 50. And to generate the parallel fibers, I've done the same, 7 micrometers multiplied to obtain 2 big, long axon of 291 sections. To do everything, of course, I've used Bluepieopt, in this case on the main machine that is at CSCS, with Python 2.7, Neuron 7.5, and of course, since the way that I'm using the Bluepieopt, which is not exactly standard, when usually an optimization give best individuals, I don't use them. I really don't use them. Because the best individuals are not always the best. You can place the best feature on the planet, you can have the best traces, but it's not so simple to obtain the best as the best. So we use another way, is to simulate all the last generation. So if I have 100 individuals, I do all of them to see if there is something better or worse in many cases.
How do you decide if it's something better?
I have to validate them. That's the tricky part, because when I do the optimization, I obtain a generation, the last generation, so maybe 100 individuals. So I have to check them, I have to simulate them, check them for specific properties. For example, in the case of the granule cell, I use as we will see in the last, I use the amplitude of the spikes in the axon. So that's a reference point, because as you will see in the last slide, the axon is there. I've placed it there. It must be working. If not, it's a waste of time and computational power. So in this case, for example, I have to use different section lists. So I have an ILOC, initial segment, ascending axon, and parallel fiber, because the sodium channels, I have 2 different sodium channels based on experiments. So with FHF in the first 2 sections, the rest of the axon doesn't have this protein. So there is completely a separation of these 2 sodium channels. And of course, the other channels are placed, for example, the potassium, the main KV 3.4, or calcium channels, or more 95% of them are recorded from actual granule cells, so it's not taken from other cells, or from granule cells. So I have a template, that is the upper trace, which I used to obtain resting voltage, AP width, HP depth, and so on, in frequency, adaptation index. So all the features that I have to use in the actual optimization with all the possible combinations. So 10 ampere, 16 picoampere, 22, etc. And based on a previous question, in fact, I use a few individuals, or at least in this case it was 64 individuals for each optimization. In the last I went around 200. In the case of Purkinje cell I was around 300. But I don't usually use more than 15 generation, because I have seen that too many generation, they can collapse. After a while, maybe some specific character became like a genetic collapse. So instead of finding good results, the worst possible results start to become the normality. So I had to reduce the number of generation. Of course, if I already know more or less how the channels are placed, the ranges are small, it takes only 6 or 7 generations to reach steady state, because this is the final version, so it's the final optimization, not something that before this one there was another one. I had to change the ranges, I had to modify them, I have to do another optimization, and so on. So this is the last version. Now, to check if everything is correct, I have to check the concept of the axon. So using the same section list, I defined the recording location. So in this case, I record at the same time from the soma, initial segment, the first section of the parallel fiber, and the last section of the parallel fiber. So it's very straightforward to add other recording sites to Bluepieopt. And all of these for each step, so at each current injection. And the results is this one. So I can see soma, initial segment, ascending axon, parallel fiber. The version above, if I only see the soma, I can say, okay, it's not exactly the best granule cell on the planet, but maybe it's working. Okay, the last part of the trace is an indication that the cell is not working right. But for Bluepieopt, that is a working cell. It was obtained, optimized, and so on. But if I go and look at the axon is dead. So this is not a working neuron. This is something that it doesn't exist. Instead, the other one, you can see that the soma has a specific shape, and these spikes are sent through the axon with a specific pace. Amplitude is different as it has to be, etc. So to conclude, the pipeline is the same as the one already shown before. So you start with the morphology, you choose which are the most important features you want to find in your model, so spike height, frequency, spike counts, and so on. Experimental traces to have actual data to do the optimization. In my case, I used the information from 3 different current injection for the Purkinje cell and 3 different current injection for the granule cells. The number of the individual for generation, the total number of generation, it depends on if you have already done many other optimization, you already know more or less which are the good ways. If not, you have to do more individuals, more generation, or more optimization, and so on. So this is only the final part. There is a job before. And since I'm not using the best individuals, I had to take and simulate the entire generation with a modified Bluepieopt template. I've added MPI code so I can run it on a cluster without problem. So I can do the optimization and the simulation directly on the same machine. And so I can see how each of them works. And then I have an automatically Python script for the Purkinje cell and for the granule cells that takes in account specific parameters, for example, the presence of the spikes in the axon or the frequency in the soma, and automatically decide if it has to be taken or rejected. So, for example, in the parallel fiber, I take the amplitude. One can take the frequency. One can choose the feature that more likes, or the firing frequency during the current injection in the case of the Purkinje cell. And this is the end.